and talk a bit about alignments now. You have seen some small alignments already today. These are essentially slices of a larger whole genome alignment run. And we do these, so these are all pre-run and not calculated on the fly. We either have pairwise alignment between two species or multiple alignments between larger sets. You can use these to identify highly conserved regions. These are related to sequences that evolve more slowly, and we know that the highly conserved regions, they tend to be important, functionally important. However, we also find highly conserved regions in non-coding regions, and we're sometimes not quite sure what that means yet. You can also use whole genome alignments to look at regions where you think that there may be a problem with that region, that the gene annotation looks a bit dodgy or something like that. And you can check this with an alignment. And finally, they are used to define syntenic regions as well. Our alignments are run with blast Z or last Z if they are pairwise alignments. For the multiple alignments, we either run EPO analysis or Mercator-Pekin analysis. The EPO analysis is done for sets of species that are relatively closely related, and they are listed here. So these are the alignments you have and that you can choose from. For the amniota, which include the mammals and the birds, as a set of 51 species, we run the Mercator-Pekin analysis, and this is because it's a bit more forgiving when it comes to differences between the genomes there. Questions on alignments? No questions on alignments? Okay. I'm not familiar with the details of this analysis. It's an alignment algorithm that we have not invented, somebody else has, and we are using this as this is the most, as I said, relaxed one when it comes to differences. It is, yeah, you can go in there. Yeah. And I can go as well. More questions? Okay, let's have a look at this then. In this demo, we are looking at a region on chromosome 2 that contains the HOX-D cluster, which is important for lymph development, and we're going to look at alignments and conserved regions there. And that's on page 53 to 56. I will copy this region. I will go back to my home page and select human, paste it in, and now I'm again in my location tab, where you can see this cluster of HOX-D genes here in human. What I'm going to do is I will focus on this third picture, the detailed view here, and I will add a couple of alignment tracks now. So for this, I'll go to configure this page. Then I'll do a few things at once. I'll go to the alignments section here, blast Z and last Z alignments. And I will add alignments for some species. I will add chicken and chimpanzee and mouse. Okay, so this is our pairwise alignments. And now I will add some tracks for conserved regions as well, under conservation regions. There's one which is already pre-selected here for constrained elements, and I will add the one for the conservation scores just above that too. And then I will close this menu. And have a look at the result. So let's start at the top. We have two tracks there in pink now, which are related to conserved regions. This one here, with the positive and the negative scores. This is raw data signals related to conservation of individual nucleotides there. So positive means conserved, negative means not conserved. And these raw data scores were then used to annotate constrained elements. These are represented as these blocks in the track above. So at each point where these scores reach a certain cutoff, we annotated one of these constrained elements. And they're called constrained elements because they are considered to be constrained by evolution. And what you can hopefully see looking at the pattern of these scores and the elements and how they line up to the axons is that the coding regions, say here, are more conserved than the non-coding regions. Yeah, it gets a bit busy with HOXD by now, but yeah, so here's the one axon here where you have that peak and then the constraint element here, for example. Okay, so these are the conserved regions, and then I added alignment tracks as well, which are shown at the bottom of the page. Again in pink. I'm not sure why all of this is in pink. So here, a pink bar represents an alignment of that species with human, and white is no alignment. And you can see that, of course, for closely related species, chimpanzee and human, who have more alignment than for, say, chicken and human. Okay, you can also look at alignments for these different sets I mentioned, or for pairwise alignments. For this, you go to Alignments text in the menu on the left, and you have to select your alignment. So if you click on the Select Other Alignment button, you would start here. So with all alignments, and you can choose multiple or pairwise. I will do a pairwise alignment with mouse and human now. So I will click through pairwise and then rodents, rats and mice to the mouse reference genome and select that. What I've selected is shown on the right hand side, and now I can click apply. And you can see that this region between human and mouse has an alignment, and it comes in 4 different blocks in this table here, of which one is the largest, and this aligns human chromosome 2 with mouse chromosome 2. So this is most likely the most reliable one. So I can click on block 1 here. and I get a preview of this alignment in this region between human and mouse. I can show everything, but I won't be doing this now because it's large. But you can of course download it in a variety of formats as well. Yeah, I show you one more thing, and that is how you can compare alignments and their in a wider region and the annotation in that region. So for this you go to Region Overview in the menu on the left. Oh, sorry, no, Region Comparison, just under Alignments text. And again, you need to select the species you want to look at. I'll go for human and mouse again. So rodents, rats and mice, mouse. And now you have your reference genomes of human and mouse shown next to each other, underneath each other, and then further down at the bottom of the page you get a more detailed view where red is human and this here is mouse, and then these pink bars represent the alignment. They're connected by green blocks, and you can see there are some gaps in between human and mouse in the alignment, and also the gene annotation in that region is quite different. I think that's what I wanted to show here for the alignments. Questions on this? No questions on this? Okay. Does anybody have a strong desire to do an exercise for this now? Yeah? Because we have one more module to cover after this, which is Biomart for data export, and it would be quite important that we do this today. So I don't want to keep you longer than you wanted to be here. One exercise, short exercise, yeah? Pick one of those. They are in your question book on page 15 to 16. We have some for trees, some for alignments, protein alignments, and also taxonomic compara, which will be interesting for people who work on bacteria and other non-vertebrates. Alright, has everybody had time to finish one exercise? Who wants more time to finish one exercise? A little bit? Yes. Okay. Can we move on? Before we move on, are there questions on these exercises? Do you want me to show anything? Anything unclear? Questions? Okay. So now we're going to spend the last, before the summary section, we're going to have a short look at how you can export data from Ensembl in larger sets. This is done with a tool which is called Biomart. I will talk you through it and the 4 steps that you need to use for this tool, and then also mention the R package that is available for it. What is this tool, Biomart? It's a tool in your browser, just as the web. You can use it to generate custom data tables with the data of your choice and sequence files. You don't need any programming skills for it. You can just do it in the browser with your mouse clicks. Why would you use it? You would use it for certain tasks which become too complicated and too time-consuming in the browser. So, for example, you could download sequences for 100 genes at once using Biomart. If you were to do that in the browser, you would have to go to every single gene page and click download sequence. Also, it can query multiple things in our database at once, which makes it quite useful for ID conversions. So you can convert an Ensembl ID into an HGNC symbol, into a RefSeq ID, and so on. And this is available for medium to large amounts of data, like hundreds or maybe 1,000 entries, but not huge amounts, for which we have other access methods. You can find this tool in the top header of your browser, in Ensembl and in Ensembl Genomes, and also in the Tools section. It is available for all species in Ensembl and many species in Ensembl Genomes, except for bacteria. I'm sorry about that. This is because we have 44,000 strains in Ensembl Genomes for bacteria. These are the 4 steps that you need to use with Biomart. In the first step, the dataset, you choose your database and your species. In the second, you need to narrow down your dataset to make it accessible and workable to a web interface. And then the third step, the attributes, you specify what you want to see in your output. And in the last step, the results, you preview the results and then export them either as a table or as cluster sequences. To go through this in a bit more detail, so in the data set you need to define your species, and then your database. We have databases for genes, for variation and for regulation. It's very important that you choose the correct one. If you choose the wrong one to start with, you will get stuck later. There's a specific case for mice. If you work with a mouse, you need to select mouse as your species and then your mouse strain as the database, because we have 16 of these. In the second step, you then need to apply filters to narrow down your data set and make it smaller. And you can combine a variety of different filters, and what you will get as the output is the intersection of these filters, and this is what we call attributes, your output. As an example, you could say you apply two filters. You would go for filter 1, which are genes on chromosome 2, and filter 2, genes which are associated with heart failure. As your attributes, you would get all genes that are on chromosome 2 and at the same time associated with heart failure. So this intersection here. And then for the attributes, your output, you have to carefully think about what you want. And these come in different sections. You have got IDs, features, variants, homologs, and sequences you can choose from. And in the last step, the results step, you preview your results, and then you can download it in HTML in different table formats that you can open in Excel, and finally as sequences in FASTA format as well. For the people who are programmers and interested in larger scale access, there's also a bioconductor package on this if you are interested in it. I will leave it there for bioconductor. And there's a publication on this. Two, actually, questions. So just a short presentation. No questions? Okay. It is straightforward to use, I would say at least. So what we do in this demo now is we will get data for a set of 6 human genes which have these names here, and we will find out for 6 of them at the same time. There are Ensembl gene IDs, their functions via their Go terms, and also their cDNA sequences. And this is of course only an example. You could do this for several hundreds of genes at the same time. The way we need to set up this biomarker query is as follows. Our data set is human genes. Our filters are these 6 gene names. Our attributes are the Ensembl gene IDs, the Go terms, and the cDNA sequences. And then we get the results. And yeah, this demo is on page 57 to 61 of your course book. I'll leave my presentation. And I will go to my training page again. And this copy sheet, plain text file, to copy these 6 gene names here for the demo. Okay, and then I go to Biomart, either from the top header or in my tools section. And this is how the empty interface looks like. We need to choose our database first. In this case, this is Ensembl genes. And we select our species as the data set. This is human genes. Now we have done this basic choice. You will see that on the left-hand side, this menu populates with filters and attributes. There we go. And now we can go into filters to make this data set smaller. So I click on filters. And you can see there are many options for filters. You can expand all of these. You can filter for regions, genes, phenotypes, and multiple things at once. I will filter for gene symbols, so I expand gene here. And my gene names are actually external references. So I will select this box here, input external references ID list, and paste my gene names here. In this same box, Biomart will not be able to read this without you telling it what these are. So you need to select from the drop-down menu the correct option. And you can see there are lots of different IDs we support. I know that these are HGNC symbols. This is the major source of our gene names. So I select that, but they all come with examples in brackets. You can, of course, upload a file here as well. So now I've selected my filters. I do a count, so I click on count at the top left to see if these have read correct. And if they have, then I should have 6 genes. Yeah, so now my data set is 6 genes. You can see this here. And now I can go into attributes and specify what I want to see in my output table. At the top you have different options ranging from features to sequences. I will stay in features for the moment. And I said I'm interested in Go terms and NCBI gene IDs. These are both under external because they come from other databases. So I expand external and select the Go term accession name and definition. And then under external references, the gene IDs, NCBI gene IDs. You can see everything that I've selected on the left-hand side, plus gene stable ID and transcript stable ID, which are already pre-selected. I will add my gene names to those as well. So I'll go into gene, and I will add gene names here. And now I can do a final check on the left if I have everything, and just click on results. And this gives me a preview of 10 rows of my output table with all the different things that I exported for these genes from the database. You can view all of this if you like, but you're more likely to want to select one of these formats, Excel, for example, and click Go to download them. If it's a really big data query, you could also put in your email address here, and we email you when your results are ready to be downloaded. So that was the data in table format, and we can now have a look at how we get the sequences for these 6 genes as well. They obviously don't come in table format, they come in FASTA files, but instead of doing all of this again, I will just go back into my attributes in the menu on the left, and then select sequences at the top. For the sequences, you have 2 sections. If you expand header information, you can see and choose what you want to see in your header. Gene stable ID and transcript stable ID again are already pre-selected. I will add my gene name as well. And then in sequences, you have a little graphic which shows you in red what type of sequences you will get, and you can select that and it changes. And we said we are interested in cDNA sequences, so I will select those. And now just click on results again. And now you can see a preview of this big FASTA file, which includes all transcripts for all of these 6 genes. So here's one transcript for this gene, here's another transcript for another gene, and so on. So yeah, that's how you get tabular data and sequences with Biomart. I hope this is useful. and saves a bit of time for you. Any questions on this? Do you want to do 1 or 2 exercises? Yeah. There are quite a few exercises for this, which are on page 17 to 19 of your course book. Just to get different types of data with a biomark from Ensembl. Please see what you're most interested in. Some are related to genes, some are related to variants, others to comparative genomics. And yeah, please do 1 or 2 exercises. For these exercises, you definitely want to use this plain text file here, which has lists of IDs you need. So you can't copy these from the PDF, and typing obviously takes a long time. So yeah, if you want to try a couple of these, please do. And we spend about 10 minutes of this, and then I summarize and point you to further online help and documentation for all the things that we have not done today. Okay, I think that would have been enough time for everybody to do at least one exercise. Are there questions on the exercises? Are you happy to look at the answer books yourself? Okay, perfect. So now we don't do this. I will point you to resources to this later. If you could spend the next 5 minutes or so, please, to fill in the feedback survey for us. We run a lot of courses like this as a team all over the world, and we try to constantly improve on those. So some feedback would be really helpful. You can find this survey on today's training page, just at the bottom. It's a Google form. If you could fill it in, that would be great. And once you've done that, I will summarize and show you to all the online stuff as well. I just asked everybody to fill in the feedback survey, which is at the bottom of the page. You could fill in asking your colleagues, that would be great. You need to open the thing called the browser or Kindle. I just asked everybody to fill in the feedback survey, which is on the training page. That would be great. Okay, I think I can start wrapping up. Okay, to summarize today, I hope you have all learned that Ensembl is a genome browser that integrates different types of data, genome variation, annotation, comparative genomics, and also regulation, which we have not done today. This data is organized in a variety of ways. We have been mainly using the website today. There are archive sites. There's a GRCH37 archive. There's an Ensembl genome site. You can mine this data and export it with Biomart. We have also underlying databases, MySQL databases that you can access with APIs. This is advanced access for people who can program. These databases are also on the FTP server, which is public. This is the all-important slide for today. So if you take one thing home, I'd suggest it is this slide. That lists all the different resources online where you can find further help and documentation. You have seen our training page already. We have tutorials on our website, but we are also part of the European Bioinformatics Institute, which is EBI at ebi.ac.uk. And one of the pillars of the EBI actually is training, providing bioinformatics training for free to the scientific community. This is their website. This is the training section just here, where all the courses they do are listed, but there's also a train online section here. And this is a very valuable resource for free bioinformatics online training. They're really hands-on as well. You can filter by different topics here. This is in alphabetical order. I will go straight to the page 3 though, where there's Ensembl material. So we have quite a bit on there. And our courses on there range from short webinars on certain topics such as LD to a 3-hour REST API course if you're interested in that kind of thing. I would like to point out this Ensembl browser webinar series here. This was recorded last year as part of a distance learning program with Columbia actually, and so because it was recorded last year, it's pretty much up to date, and this covers everything that we do in a browser workshop such as this, plus the modules that we have not covered, and they are all organized with in the same way as today, so presentation, demo exercises with videos. We have today done all of this. We have not done regulation, which you can find here. We have not done data upload and the REST API either. So this is where you can continue to learn yourself. So have a look at that. Yeah. We have a YouTube channel, which I mentioned earlier. This includes small videos of a few minutes, just some tutorials, and then larger, longer webinars up to an hour. You can email us at any time at helpdesk@ensemble.org, and a member of the outreach team will answer your questions. You can join one of our public mailing lists. There's a developers mailing list for large-scale access and programmatic access. And there's an announced mailing list where we talk about new data, new releases, and what we plan for our releases. We have social media accounts, Facebook, Twitter, and a blog. And the blog is also the place to go if you are interested in working with us, or for us rather. We do write publications. You can find the publications on the website shown at the top. Please do cite us if you use this data. We provide this for free, and it's very important for our funders that we get citations as well. And this is us. Last year, in 2018, we are the biggest group in VBI. At any time, around 60 people. And there are only 4 outreach people who deliver workshops like this and do represent all the others and their hard work. And that brings me to the last slide, which is the acknowledgement. I'd like to thank this entire team, Ensembl team, our funders. Our main funding comes from the Wellcome Trust, and they make it possible as well that I'm here today. And we get co-funding from other parts, other funding agencies, including the European Union. And finally, thanks to the organizers, to the local host, and to all of you for coming today. I hope this was helpful. Thanks. Okay, that was it. Thank you. Yeah, that went really well. Good. Thank you.